Good afternoon, student. Good afternoon, everyone. In the last class, we were studying about pollination, that is, the transfer of the pollen grains from the anther to the stigma of the same flower or the another flower. Based upon the source of the pollen, that is, whether the pollination takes place within the same flower or within a different flower or within the same flower of the same plant. Depending upon that, there are pollination is of three types. One is autogamy, what is another one is Gaitanogami, and the third one is Xenogamy. Now, what is this autogamy, Gaitanogamy, and Xenogamy? Autogamy is strictly self-pollination process. It is a pollination which takes place within the same flower. The pollination which takes place within the same flower, that is the transfer of the pollen grain from the anther to the stigma, takes place within the same flower. That is strictly self-pollination process. Whereas the second one is Gaitanogamy, where this is considered also as a kind of a self-pollination, where what happens over there? The anther, that is the pollen grains, move or transfer to the stigmatic surface of the flower, but within the same plant. Whereas xenogamy is a cross-pollination process. Now, what is this cross-pollination? Cross-pollination is nothing but where the transfer of the pollen grain from the anther to the stigma takes place between two different flowers of the two different plants, maybe within the same species. So there are three types. So first you have to remember autogamy, Gaitanogamy, and Xenogamy. The first one, that is autogamy. So here you need to remember the definition. Once again, I'm repeating, in this chapter, the definitions are very important and the types. In autogamy, which I said you already, it is a self-pollination process where the transfer of the pollen grain from the anther to the stigma takes place within the same flower. There are two types of conditions in self-pollination process. There are two types of condition in this self-pollination process. First thing is, self-pollination requires closeness of the stigma and the anther. Yes. So what is needed here? Probably the stigma and the anther has to be very closer so that it is easy for the transfer of the pollen grain from the anther to the stigma surface. You all know the flower. Yes. So now imagine a hibiscus flower. You all know how the anther and the stigma are closely related there. Yes, how they are closely been closely designed over there, how so that the anther or the pollen grain can move on to the stigmatic surface. So the closeness of the stigma and anther is one of the conditions. Based upon that, there are two types of flowers. How many types of flowers? Two types of flowers. One is chasmogamous flower, another one is cleistogamous flower. Okay, so what is the difference here? So can you see here the flower? Here it is a self-pollination takes place. How self-pollination takes place? That is, the transfer of the pollen grains easily move on to the stigmatic surface of the flower. Yes, so here the self-pollination takes place. In according to these conditions, there are two types of flower. One is chasmogamous, one is cleistogamous. Now, what is this chasmogamous flower? Chasmogamous flowers are those flowers with the exposed anthers and stigma, but they should lie close to each other. Yes, so here what happens? Here the exposed anthers. Anther means the male reproductive part is very exposed. Okay, exposed means it is very exposed to the very exposed in the flower. You can easily make out or you can easily see the anther there, whereas the stigma also, because so that the transfer of the pollen grains can be done easily, but they lie close to each other. Like for the example in hibiscus, you have seen in hibiscus flower, the anther and the stigma, the pollen grains are very distinct there. Yes. So once again, I'm telling you to imagine a hibiscus flower. So when you imagine one hibiscus flower, you can see the pollen grains very distinct over there, and also the stigmatic surface. Yes, the red color stigmatic surface, when you see it in the style of the hibiscus flower, those are very distinct, means they are very exposed, where the transfer of the pollen grains can be done very easily, and self-pollination can take place. Yes, the self-pollination can take place. Whereas second one is cleistogamous plant. So these two, Chasmogamous and Cleistogamous flowers are important. So remember why it is important. Differentiation between Chasmogamous and Cleistogamous flowers can be asked for 2 marks. Okay, the differentiation between Chasmogamous flower and Cleistogamous flower can be asked for 2 marks. When you are writing about the Chasmogamous flower and Cleistogamous flower, you both the places you need to give examples. Okay, both the places you need to give examples, so that 1 point you can write, whereas second one, your second point can be written as an example. Clear? Even it is asked for 2 marks, you need to remember a Chasmogamous flower, one definition you need to give and one example. Whereas in the Cleistogamous flowers also you have to be doing the same. You need to give one definition and the example. Now coming to Cleistogamous. Now what are these flowers? Cleistogamous flowers which do not open at all. Those flowers remain closed all the time. Okay, they are the buds. Buds you have seen, right? The buds, those petals are closed, whereas the sepal close them. Yes, the sepal part, the green part, close them. And these flowers are also the same. Cleistogamous flowers are also the same, where flowers do not open at all. Now, Cleistogamy is a disadvantageous because there is no chance of variation. Yes, because there is no chance of adulteration, or there is no chance of contamination of these flowers. So no other pollen grain can come and pollinate them. No other pollen grain can pollinate them. So it is disadvantageous for variation, because there is no variation seen, no genetic variation seen at all in these flowers. Why it is not seen? Because they remain closed all the time, and the pollination takes place within the closed flower. The pollination takes place within the closed flower. That is, the transfer of the pollen grain from the anther to the stigmatic surface takes place within the closed flower. Okay, so there is one disadvantage is that there is no variation at all. Whereas in the other plants, such as like in the hibiscus or any other paddy, wheat, or any such plants, annual or biennial plants, there is the chance of variation. Why? Because they have got exposed anthers and stigma. Any pollen grain can come and pollinate the stigmatic surface of such flowers. Yes, they can come and pollinate the stigmatic surface of such flowers. Whereas in Cleistogamous flower, the flowers do not open at all, and there is this disadvantage. There is no variation, no genetic variation seen. Example is oxalis and viola. Okay, oxalis and viola. Now you will see which are these flowers. The viola, this purple color flower you must have seen, right? Yes, the purple color flower which you see among the grasses, very small flower, and this commelina as well. The commelina flower, which is you see the blue color flower here, you see among the grasses or the weeds most of the time. So such flowers do not open. Okay, such flowers do not open, and they pollinate when they are closed. They pollinate when they are closed. Now, Chasmogamous flower and Cleistogamous flower. Some plant produces 2 types of flowers. Example, that is viola, oxalis, and commelina. Flowers with exposed anthers and stigma are called as chasmogamous. Do not get confused. Those flowers which have got anther and stigma which are exposed are known as chasmogamous. Whereas the flowers which do not open at all, remain closed all the time, and the pollination takes place within them when they are closed, are known as cleistogamous flower. What are they known as? Cleistogamous flower. And differentiation is important, students. Examples are important. Viola, commelina, and oxalis. You need to remember this names. Okay, viola, commelina, and oxalis, you need to remember these names. Now, also you must have seen these flowers in the weeds, among the grasses, in fields and all, or anywhere where the weeds are growing. You must have seen this purple color flowers. These are known as viola and commelina flowers, which are known as cleistogamous, which do not open at all, and they do this pollination by the self pollination process. That is strictly autogamy. It is, that is strictly autogamy. There is no chance of cross pollination in this flowers, that is cleistogamous flower. So if it is asked for you to write about autogamy, that is self pollination, first you need to give the definition. What is autogamy? That is transfer of pollen grain from the anther to the stigma of the same flower. You need to give the definition. If it is asked again to explain about the autogamy, you need to write about the conditions. So when you write about the conditions, you need to give chasmogamous flowers and cleistogamous flower. Now, what is chasmogamous flower? You need to define it, give an example, cleistogamous flower, definition and example. Clear? Now, why examples are important? When you are writing differentiation, there should be 2 points for 2 marks. You are understanding it? 2 points for 2 marks. One definition, one example. Clear? So chasmogamous flower, flowers with the exposed anther. Exposed means which are exposed, which are very much open to the environment or to the surrounding. Clear? Second one is cleistogamous flower, which do not open at all. They remain closed all the time and pollination takes place there. One of the disadvantage is this: there is no chance of variation, means no other pollen grain can pollinate it. Next, coming to Gaitanogamy. Now, what is this Gaitanogamy? Gaitanogamy is also a self-pollination process, students. You need to remember it. That is also a kind of a self-pollination process. Okay, it is also a kind of self-pollination process. Gaitanogamy is also a kind of self-pollination process, and you need to remember this. Autogamy is also a self-pollination process. Gaitanogamy is also a self-pollination process. Transfer of pollen grain from anther to the stigma of the another flower of the same plant. Clear? Now you need to remember the definition. It doesn't take place within the same flower, students, but the flower which is present in the same plant. Now this is one plant. Okay, forget this. This is one plant. There are 2 flowers in one plant. Yes, there are 2 flowers in one plant. So what happens? The pollen grain from this plant or this flower, here, the pollen grain from this flower will pollinate this flower. Clear? The pollen grain from this flower will pollinate this flower within the same plant. Both the flowers are in the same plant, but the pollination takes place from this flower, that is the transfer of the pollen grains takes place from this flower to this flower. Okay, so it is known as Gaitanogamy, and it is one of the self-pollination process. That is the transfer of the pollen grain from the anther to the stigma of the another flower of the same plant. Genetically similar to autogamy. Example is cucurbits. Cucurbits, that is of the cucumber family, that pumpkin family. So thus pumpkin and all cucurbit family, this Gaitanogamy takes place. That is the transfer of the pollen grain from the anther to the stigma of the another flower of the same plant. Transfer of pollen grain from the anther to the stigma of another flower of the same plant. And it is genetically similar to autogamy. It is genetically similar to self-pollination process. Okay, genetically similar means it can that the transfer of the pollen grain from this flower to this flower, but within the same plant. From this flower to this flower, but within the same plant. Okay, so Gaitanogamy is the transfer of the pollen grain from anther to the stigma of the another flower of the same plant. Okay, there is one plant. It has got two flowers. So the flower one, from there the pollen grain will come and pollinate the flower two. Okay, so this is a kind of a self-pollination process. Example is cucurbits. Example is cucurbits. Next, coming to xenogamy. Now, what is this xenogamy? Xenogamy is nothing but a clear cross-pollination process. You need to remember, xenogamy is nothing but a cross-pollination process. Now, what do you mean by this? Cross-pollination process. Pollination takes place between the two flowers. Pollination takes place between two flowers born on the two different plants of the same species. Pollination takes place between two flowers born on the two different plants of the same species. Now, what happens here? See, this is one plant. This is another plant. For example, if I take, it is a papaya plant 1, papaya plant 2. Now, what happens here? The anther from this flower. The anther is that the pollen grains from this flower go and pollinate the other flower of the another papaya plant. That is papaya plant 2. The pollen grains from papaya plant 1 pollinate the other papaya plant 2 flower. Okay, pollinate the papaya plant 2 flower. So what happens here? There is the cross-pollination. Yes or no? Cross-pollination. Why cross-pollination? Because the pollination is not taking place within the same plant. The pollination is that the transfer of the pollen grain is taking place between the two different plants, but of the same species. That is the papaya plant. This is papaya plant 1, papaya plant 2. This is what I have given you the example. Now what happens here? The pollen grains from the papaya plant 1 go and pollinate the papaya plant 2. Yes, they pollinate the papaya plant 2. That is the transfer of the pollen grain from the anther to the stigma of the different flower, but of the same species. And this process is known as cross-pollination. Why cross-pollination? Because the pollination is taking place between the two different plants. Yes, between the two different flowers of the different plants. Between the two flowers born on the different plants. Here, the pollination takes place between two flowers born on the two different plants of the same species. Same species, I already said you, papaya. This is papaya plant 1, papaya plant 2. Here, the anther is that the pollen grains go and pollinate the stigma of the another flower, but of the same species. So this process is known as xenogamy. Genetically different pollen grains are brought to the stigma, thus introduces genetic variation. Yes, your genetic variation can take place in a cross-pollination process. Why? Because different pollen grains can come and pollinate. Different pollen grains can come and pollinate these plants. So there is a wide possibility of genetic variation. Why there is a wide possibility of genetic variation? Because the pollen grains from different species also can pollinate the different plants. Okay, they also can pollinate the different plants. So here pollination takes place between two flowers born on the different plants. Here they are born on the different plants but of the same species. Now you'll ask me why you have taken the example of papaya plant. So here the very easy answer is papaya plant is monoecious. So what happens here? It is either male plant or female plant. Okay, either male plant or female plant it is. Here only the male flowers are grown, and in another plant the female flowers are born. Both the flowers are not born within the same plant. Okay, both the flowers are not born on the same plant. One plant will have only male flowers, other plant will have only the female flowers. So the transfer of the pollen grain from the stigmatic surface takes place by the cross-pollination process. It takes place by the cross-pollination process. And because there is this possibility of cross-pollination, there is a wide possibility of genetic variation. Means any pollen grain can come, or any pollen grain can pollinate that plant, and there can be a genetic variation. A different variety can be grown. A different variety can be grown. So there is a wide possibility of genetic variation in xenogamy. Whereas, remember it, do not get confused. Whereas in cleistogamous flower, there is no possibility of genetic variation. Why? There is no possibility of genetic variation because the flower do not open at all. The pollination takes place within the closed flower, and there is no possibility of the contamination or adulteration of the pollen grains. Whereas in cross pollination, there is a high chances or high possibility of the variation taking place between the two plants. So here in cross pollination exam, a very easy and apt example is papaya plant. Why? Because the male flowers and female flowers are grown in a different plants. So as we have studied about the pollination, we studied about the definition of the pollination, which is very important. Pollination definition, types of the pollination, autogamy, Gaitanogamy, Xenogamy. In autogamy, the conditions and the two different flowers, and difference between chasmogamous and cleistogamous flower important. What is Gaitanogamy? Definition is important because the differentiation between autogamy, Gaitanogamy can be asked. Differentiation between Gaitanogamy, Xenogamy can be asked. So the differentiation can be asked for the 2 marks or 3 marks possibly in your examination paper. So it is very important. And it has been asked time and again also, the difference between Xenogamy, Autogamy and Gaitanogamy. So it is very important for you to know the examples and the definitions. When we are studying about the pollination, there are agents of pollination also. Now, when we only focus on the definition of pollination, let me focus on the definition of pollination only. The pollination is nothing but the transfer of the pollen grain from the anther to the stigma. Yes, some take place within the same flower, but some of those cross-pollination process needs one mediator. What do you mean by a mediator? Mediator is nothing but a medium from where the pollen grains can be transferred to the stigmatic surface. The pollen grains can be transferred to the stigmatic surface. So there are some medium or the mediators, and those mediators are known as agents. Those mediators are known as agents, which helps in this process known as pollination, which helps in this process, that is to transfer the pollen grains from the anther to the stigma. So there are 2 types of agents, students. One is abiotic agents and one is biotic agent. Now, what is this abiotic agents? Abiotic agents are nothing but a non-living agents. So you need to remember abiotic is nothing but non-living agent. So it is a non-living agent, whereas the biotic agents are living agents, which are the living, moving agents. Whereas in abiotic agents, those are non-living agents. So this question can be asked for you. Write down the agents of pollination, or give the abiotic agents of pollination, or give the abiotic agents of pollination. So it's one of the important question. In abiotic agent, that is the non-living agent, which are those 2 types? There are 2 types, that is one is wind and one other one is water. So remember, abiotic agent or non-living agent, there are 2 types, wind and water. And biotic agents, there are insects, birds, bats, reptiles, and mammals. Insects, birds, bats, reptiles, and mammals. And biotic agent, that is in living agents, these are the different agents which helps in the pollination. Now, depending upon this, depending upon the biotic agents, whether it is about insects, whether it is about birds, mammals, or reptiles, depending upon that, the pollinating agents are of different types. We will be studying it in the further slides. That is, either it can be zoophily, anemophily, hydrophily. So depending upon that, we will be studying. So you need to remember there are two types of agents: abiotic agents, biotic agents. Abiotic agents are what? Those agents which are non-living. Biotic agents are those which are living. In abiotic agents, there are two types: wind and water. In biotic agents, there are insects, birds, bats, mammals, and reptiles. So here, in abiotic agents, abiotic agent pollination, there are some of the adaptations which are taken up by the flowers. There are some of the adaptations taken up by the flowers. Now, what is this adaptation? The adaptation is nothing but so that these flowers have adapted themselves so that it becomes easy for the pollination to take place. So that it becomes easy for the pollination to take place. So one among them is, first one is wind pollination. Wind pollination is known as anemophily. Wind pollination is known as anemophily. Now, do not get confused. Anemophily is nothing but wind pollination process. It is a wind pollination process, and anemophily is not a animal pollination. Remember this. It's a wind pollination. Always remember, anemophily means wind pollination. It is not an animal. Okay, animal is zoophily. Wind pollination is anemophily. Next is about the water pollination, hydrophily, and there are some of the adaptations taken up by these plants which we will be studying in the later classes. So till here you need to remember there are some of the adaptation in flowers for the pollination to take place. Today we studied about the pollination, definition of pollination, then the types of pollination. And types of pollination, we studied about autogamy, Gaitanogami, and Xenogamy. Then later we studied about the agents of the pollination. Yes, pollinating agents we studied. There are three types. There are two types: abiotic agents and biotic agents. In abiotic agents, that is non-living agents, we studied about wind and water. And in biotic agents, insect, bird, bats, reptiles, and mammals. If you have any doubts, any confusion, anything you want to ask me, any of the doubts you want me to you want to clear, or any question, any concept you have not understood, you can ask me personally so that I can explain it to you. Anything, any kind of any confusion is there, you can just contact me so that it will be easy for you and for me as well, so that we can talk it out and clear your doubts. Okay, thank you. Have a nice day, students.